Hello everyone. Today I am going to take class on a topic morphology of cell injury, necrosis and gangrene. As we know, cell injury are of two types, reversible and irreversible. The morphology of reversible cell injury includes cellular swelling and fatty change, whereas irreversible cell injury includes necrosis and apoptosis. Let's move on to the cellular swelling. It is also called hydropic change or vacuolar degeneration. It is commonly seen in cell injury associated with increased permeability of plasma membrane. Gross appearance, it is of increased weight, increased organ weight and increased turgor pressure, and it causes pallor. Pallor is as a result of compression of capillaries. Microscopically, there will be alteration of the plasma membrane, such as blebbing, blending, or distortion of microvilli. Mitochondria will be swollen and plasma lipid derivatives are also seen. Endoplasmic reticulum dilates and detachments of ribosomes and dissociation of polysomes also be there. The nucleus will be pale and you can see a clumping of chromatin. Okay, the etiology, the main cause is bacterial toxin. Then poisons, chemical burns, high fever, high administration of hypertonic glucose and saline also be there. Pathogenesis includes the impaired the regulation of sodium potassium pump, means the sodium potassium pumps gets failed. That's result intracellular accumulation of sodium and efflux of potassium. The rapid flow of water into the cell that will leads to cellular swelling. Okay. Now about fatty change. It is a steatosis. It is nothing but abnormal accumulation of triglycerides in parenchymal cells. The most common organ which is affected more is liver. The other organs are heart, skeleton muscle, kidney. Coming to the etiology, the fatty change in the liver may result from one of the two type of cause. They are first one is condition with excess fat, that is hyperlipidemia. It is mainly due to obesity, diabetes mellitus and congenital hyperlipidemia. And other thing is liver cell damage. The main cause is alcohol liver disease. Others are starvation, protein calorie malnutrition, chronic illness, acute fatty liver in late pregnancy, hypoxia and hepatotoxins, and Ray's syndrome. Ray's syndrome means it is due to aspirin intake. Okay. So we have to understand the normal lipid metabolism in the liver. The free fatty acid that are mainly derived either from diet of a person or from adipose tissue. Now free fatty acid enter into the hepatocytes of a liver in the form of fatty acid. This fatty acid can also be derived from the acetate in the liver. These free fatty acid that are majority they converted into triglycerides with the help of the enzyme this alpha glycerophosphate. Okay, the triglyceride then converted into lipoprotein and then it then converted into plasma lipoprotein. Okay, and also the fatty acid can be converted into cholesterol esters, phospholipids and ketone bodies for the normal body function. Okay, and now for the fatty liver there have to be a defect in the following steps. So there are 6 steps here, 1 2 3 4 5 6. Okay, so in there will be intracellular accumulation of triglycerides can occur due to the defect to one or more of the following 6 steps. The first one, there will be increased entry of free fatty acid from diet or adipose tissue into the liver. Okay, and second thing is increased synthesis of fatty acid by the liver. That is, second, at the liver more of the acetate can be converted into fatty acid. And third one, decreased conversion of fatty acid into ketone bodies resulting in increased formation of triglycerides. So there will be decreased conversion of this fatty acid into the ketone bodies in the hepatocytes. Okay, so that all the things will ultimately leads to increased concentration, increased level of this triglycerides. And fourth one is increased alpha glycerophosphate. This alpha glycerophosphate enzyme is increased, causing increased esterification of fatty acid to triglycerides. And fifth one is decreased synthesis of lipid acceptor protein resulting in decreased formation of lipoprotein from triglycerides. So the lipoprotein level will be decreased. As a result, there will be increased triglyceride level. Okay, the and also last one is the block in the excretion of lipoprotein from the liver into the plasma. That is due to the here the sixth step is blocked. That is conversion of lipoprotein to plasma lipoprotein is blocked. Okay, this all this if there is a that means if there is any defect in the following steps, that will leads to the accumulation of this triglyceride in the liver that causes fatty liver. Okay, let's move on to the morphology of this fatty changes or steatosis. That is grossly most often seen in liver and heart, and that in liver the mild fatty change not affected more, but if it is progressive, that this fatty acid get accumulated in the liver, that leads enlargement of the organ and become bright yellow and soft greasy appearance. Okay, and in heart the mainly two patterns are seen. That is first one is due to the moderate hypoxia. That is moderate hypoxia കൊണ്ട് എന്തുണ്ടാവും grossly apparent bands of yellowed myocardium alternating with bands of darker red brown uninvolved myocardium can be seen. രണ്ടാമത്തത് ഡ്യൂ ടു പ്രൊഫൗണ്ട് ഹൈപ്പോക്സി ഓർ സം ഫോം ഓഫ് മയോകാർഡൈറ്റിസ്, അതുകൊണ്ട് എന്തുണ്ടാവും യൂണിഫോംലി അഫക്റ്റഡ് മയോസൈറ്റ്സ് ക്യാൻ ബി സീൻ. ആൻഡ് മൈക്രോസ്കോപ്പിക്കലി രണ്ട് ചേഞ്ചസ് ഒക്കെ കാണാൻ പറ്റും. ഒന്ന് മൈക്രോവെസിക്കുലർ ആൻഡ് നെക്സ്റ്റ് തിങ് ഇസ് മാക്രോവെസിക്കുലർ. സോ ഇൻ മൈക്രോവെസിക്കുലർ വാക്യൂൾസ് എന്തായിരിക്കും, സ്മോൾ ആയിരിക്കും ഇനീഷ്യലി ആൻഡ് പ്രസൻറ്റ് അറൗണ്ട് ദ ന്യൂക്ലിയസ് ആയിരിക്കും. ബട്ട് ഇൻ മാക്രോവെസിക്കുലർ വാക്യൂൾസ് ആർ ബിഗ് and it is the nucleus will be pushed towards the periphery. Okay, here in this picture you can see it is the microvesicular where the vacuoles are very small. Here the macrovesicular, that is the vacuoles are very large, that is very big, and the nucleus will be pushed towards very periphery. Okay, and also the hepatocytes laden with large lipids vacuoles may rupture and it leads lipid vacuoles coalesces to form a fatty cyst.